I'd like to take the opportunity today to discuss where within FUJIFILM Diosynth Biotechnologies we're trying to develop new technologies to advance the way we make and manufacture medicines, but in a way with a sustainable perspective on that sort of technology development. So Eileen, on to the next slide, please. So just as a sort of very brief introduction, so FUJIFILM Diosynth Biotechnologies are a contract manufacturing and development organization. So we provide a range of process development, laboratory and manufacturing capabilities and services to the biopharmaceutical industry to partner and support clients, whether they're small biotech, virtual companies, academic companies, through to large pharma, in developing their processes through from early phase clinical manufacture through to commercial manufacture and all of the regulatory and quality supports associated with that. The biopharmaceutical industry market, as you can see, is growing significantly from around 186 billion US dollars in 2017 to, at the time of this reference, about $500 billion by 2025. And actually, we feel perhaps with the impact of COVID, that sort of overall number by 2025 may be much greater than that. It is a strong contributor to the UK bioeconomy, and some data that came out of the UK Bio Associates industry, the BIA, recently indicated that UK biotech companies have raised a record $2.8 billion in equity finance in 2020, in the year in the context of 2020. So it's a fantastic industry that's growing and contributing to the UK. There are a range of investments also from multinationals and large pharma companies. And actually, interestingly, those industry investments are perhaps more in the sort of research and development than they are in the manufacturing investment end of things. And from a very high level, our industry, from a technology perspective, is genetically engineering organisms or cells to produce, or in fact themselves become, biological therapies. We historically have used a largely fed batch-based process. So there's typically an upstream, whether that's running a fermenter or a bioreactor, running a bulk of material that's passed into a downstream process to purify the therapeutics to a specific requirement through a number of specific and identified batch unit operations. For the generation of clinical material, we tend to use something around the 50 to 2,000 litre sort of production scale that generates enough material to enable a client to take their molecule through the clinical trials. And we tend to try and then target for commercial scale going up to something like a 20,000 litre type bioreactor or fermenter. And the actual sort of materials of construction of those different sort of clinical trial through to commercial scale production vessels are important, and I'll revisit that later on in the slides today. So on the next slide, I really just wanted to sort of position a question really in what I was thinking about as I talked to Mark Bustard ahead of this presentation, and how can we advance biomanufacturing in a sustainable way? We know there's a real drive to growth in this industry, and I think, touching on some of Ian's introductions, there's a real drive to reducing the cost of goods of the medicines that we're making in this industry to actually sort of increase the accessibility of these sorts of therapies. So I wanted to really talk about today some technologies, some pressures on the industry, some changes in the landscape of this industry, and how we're trying to develop technologies to address those sort of challenges, but in a sustainable way. So on the next slide, I wanted to just really sort of paint a picture, a problem statement of the industry from my perspective. And really that sort of problem statement can be summarized as complexity. So we make a broad range of different complex products. These are all large molecule biologics. And as I said earlier, we're now sort of moving towards protein drugs to actually using cells or viral genes as the actual sort of therapies themselves. So there's a true complexity in the products that we're trying to manufacture. All of our manufacturing facilities in FUJIFILM Diosynth across Europe and in the US are multi-product facilities. So we offer our manufacturing capabilities to our clients and partners. And in fact, the products that we're making in our facilities change relatively regularly. And this could be up to every month. So every month, our facility needs to be making a different product. In terms of the equipment that we use to manufacture these processes, well, there are multiple technology vendors supplying this equipment. As I mentioned earlier, the traditional unit operations that we're purifying these drugs with are quite distinct, and therefore there's a range of different vendors supplying equipment to this market. And of course, we'll try and select the best equipment for the unit operation, and that therefore results in facilities across our manufacturing network which are filled with a range of different technology vendors. So this raises challenges in operator training, in the way we qualify a range of different pieces of equipment from a range of different vendors, and how we maintain them, ensure that they're ready and turned around for the next product coming along the facility asset plan. It also ranges real complexity when we talk about transferring our processes across our manufacturing network. As the client successfully navigates their molecule through the clinical trials towards commercialization, then the processes need to transfer from one facility to another as, to increase that sort of demand on material and scale of manufacturing. One of the real changes in our industry over the last decade has been the introduction of a new mode of manufacturing known as single-use or disposable or replaceable manufacturing. And I do want to talk about this in a bit of detail here today, because you may think from a conference that's talking about sustainability and reducing the environmental impact on how we manufacture, single use doesn't necessarily jump out at as a sort of strategy that would sort of lend itself to that. But I'd like to try and explore today and share some of our experiences where we do actually, in fact, it can advance the way we're manufacturing in a sustainable way. So the single-use approach is actually where we will use replaceable or disposable flow paths. All of the product contact flow paths within our manufacturing facility effectively can be delivered to the site in a gamma irradiated fashion, utilized for that process, and then disposed of after use. This reduces, as I will go on to show some data from some references in the industry, the amount of solvents, the amount of liquids, the amount of buffer requirements in the cleaning and the validation of these facilities. Now, we're not using just single-use supply chains. In fact, in our larger, more commercial scale sort of settings, we do revert to sort of the image on the right-hand side there, which is our image of one of our facilities in Denmark, where it is much more stainless steel. But for the sort of smaller, agile, clinical trial type scales, there is a number of operational advantages in single-use. One of the challenges with single use is the complexity in the supply chain, and we'll have seen more recently with the demand for such products coming through for COVID type treatments, real complexity in how that supply chain is sort of established and how it operates to enable the right single use components at the right position at the right time for utilization. One of the other areas that this industry has sort of looked across other industries, including the broader industrial biotechnology, is where we can move from a much more static fed batch mode of manufacturing to a more continuous or connected or integrated mode of manufacturing. There are some true advantages that we see within this as an industry, but we have so far not really sort of committed fully to that continuous mode of manufacturing. And I'd like to present today actually some technology development we've done in-house, which has allowed us or trying to enable us to move to a more connected and integrated manufacturing mode. But of course, in today's landscape, when we have multiple technology vendors, these sort of different vendors and equipment don't necessarily integrate and connect very well together. So again, that's prompted some internal developments to try and address that. And finally, one real specific one I wanted to call out was buffer management logistics. So we use a large quantity of buffer volumes to deliver these sorts of processes, and there's a huge logistical challenge associated with those buffers. These could be made in-house, or they could actually be sort of supplied, prepared and delivered at the point of use in the manufacturing facilities. And of course, there's a real sort of impact there on the logistics of moving large, highly dilute buffer volumes around to the manufacturing facility. So again, that's an area that we wanted to try and look at. So I guess this was just to try and paint the scene. We're trying to address these challenges that I've highlighted here to advance the way we're manufacturing our medicines, but in a sustainable way through innovation. On the next slide, you can see, how do we do this? Well, I think there's three answers really, and I think they're overlapping strategies. And I'll just spend the next sort of 10 minutes exploring technology innovation to address these. So we think if we achieve operational efficiency gains, primarily through process intensification, we can offer a more sustainable manufacturing solution. The way we design our facilities and the way that the mode of manufacturing that we execute, going from fed batch to more continuous, can again offer opportunities to leverage sustainable manufacturing. And there's a true third bullet point there is, and we're starting to do it now in terms of true cross-industry collaboration to minimize environmental impact. That's integrating into the technology vendors, the supply chain, and making decisions on the way we're manufacturing in a much more environmentally localized environment. So on the next slide, I wanted to just spend 1 minute to talk about some lessons from the past in the spirit of this section. And when we started to talk about introducing continuous manufacturing in the biologics space, I was very quickly pointed towards some experts within our organization that had done this in the past. I thought these were all great new ideas, and I had the privilege to talk down with Subhash Chowdhury, which I know a lot of you on the call will know of, and one of our very established and prestigious members of our organization, who's done continuous manufacturing in a different environment. And he very quickly shared a lot of information and knowledge in that we're already world leading in industrial biotechnology in terms of continuous processing. And he shared a case study with me using and generating a product called Protein, which was a single cell protein generated for animal feed. And a lot of the knowledge here was a precursor to towards sort of protein meat substitutes, which you've seen is a huge growing industry today. And he quickly showed me, in his experience and past, they'd actually installed in the Billingham site where I'm based, a 1.2 million litre continuous fermenter, weighing over 560 tons. And he explained how new roads had to be laid in the Billingham and the northeast area to allow this fermenter to be developed, to be delivered and installed. And actually, they ran a sterile continuous process for a record 135 days on that site. And this was a methanol feed fermenter, so some complexity in the operations. And it was a fantastically humbling conversation to talk about how we were going to revolutionize the industry moving towards continuous. And actually, we did it here on this exact site maybe 30 or 40 years ago. And there was a huge amount of operational experience gained in those sorts of histories, in sterile operations, in safety, in qualification and maintenance, and actually indeed in supply chain logistics that we've tried to leverage and build into today's new technologies. So a very humbling conversation. And on the next slide, moving to some of the technology that this is sort of influenced for us to develop. So we've developed a technology in-house called Symfonex, and this is a path that we believe is a way towards process and facility intensification, and as you'll see in a following slide, in a sort of environmentally sustainable way. So instead of having multiple pieces of equipment from multiple vendors designed to run different unit operations in the downstream, we've designed this almost as a Swiss Army knife concept. So it's multifunctional. This one piece of equipment can run all of those different multiple unit operations. So it starts to ease the pressures on training, on operation, on installation, qualification, and maintenance. We've adapted this single-use flow path concept, which allows us to have rapid facility turnaround, but significantly reduce the amount of volume of solvents required for cleaning and validating stainless steel flow paths between use. And by simplifying the flow path to one design, we start to simplify the supply chain. So we don't need to have multiple pieces of equipment with multiple different flow paths that complicate logistics, increase the amount of deliveries required on site for such sort of activities. We needed to develop software to allow us to operate this hardware. But most importantly, we developed this in a way that it could be used fed batch in today's industry processes, but also in a way that we could start to integrate and connect multiple Symfonex systems towards that sort of more continuous mode of operations, which we know can in itself lead to process intensification, facility footprint reduction, and a means forward there. So on the next slide, we've deployed these systems across a number of our sites, and most recently, just before Christmas, we deployed these systems in a proof of concept facility for continuous biomanufacturing. In fact, we had 7 of these systems deployed side by side, running a different unit operation down a monoclonal antibody purification process. And I was very pleased to say we did this in collaboration with the IBioIC, and we housed 7 or 8 of their master's industrial placement students for this proof of concept 3-month program. Very fortunately for us, some of the guys have decided to stay with Fujifilm, and they've taken up full employment with us now. But it was a great collaboration with the industry. I don't have a significant amount of time to talk about the details of this now. There is a link on the slides there, which takes you to our website, where there's some videos and there's a bit more information. But the premise here was a continuous perfusion bioreactor, which is just off camera on the left-hand side, feeding a connected and integrated number of downstream unit operations. So we were sort of processing in a sort of steady state type environment. It meant that we could generate a significant amount of material in a much smaller footprint, with a much smaller sort of infrastructure need. Again, beyond this scope of today's talk, but included to help with that sustainable position, was these systems were not only developing and delivering the unit operation, but they were able to dilute at the point of use concentrated buffers. So instead of having 1 times buffer concentrates that had to be either pre-made on site or delivered through the sort of supply chain logistics, we were able to use much more concentrated versions of these buffers that were diluted by the Symfonex system at the point of use and delivered onto the unit operation. So that reduced our sort of volume of liquids that we were managing and manipulating here significantly by an average of about 10-fold per buffer. So that was a significant volume reduction, which could knock on to reduction in manufacturing footprint, but also that sort of impact on logistics of having to deliver and ship large volumes of effectively relatively diluted salted buffered water around the manufacturing network. So in my closing sort of remarks now, on the last slide there, I just really wanted to take this as an opportunity to say, we believe that we could try and innovate and advance the way we're making manufacturings, but in a sort of sustainable way. You know, we've tried to target efficiencies in our operations through process intensification, through advancing our buffer management, and I've talked about sort of reducing bulk volumes of transport logistics, and through this single-use implementation. And many people hearing this for the first time will think, how can single-use or disposable flow paths be environmentally sustainable? And there's a lot of studies in the industry that's looked at this, and I've just taken a slide from a reference here on the right-hand side to show, in terms of total energy consumption, if you're running a stainless steel facility, which is the sort of slides on the left, you can see that there's reasonably large quantities of energy that's required for the sterilization, the cleaning, pre and post use of these stainless steel facilities. There's a small amount of energy required for the material management, material creation, and you can see that sort of total in the black there. And in comparison, we really reduce that energy requirement for sterilization and cleaning. Of course, there's a larger input to material manufacturing and delivery, but sort of all in, the sort of data here, which was sort of verified independently, and there's many more studies that can be cited. We do feel that the move towards single use can offer a path forward in a sort of sustainable way. The sort of move towards connected and continuous and integrated manufacturing allows us to rethink the way we're designing facilities. And actually, in terms of the way that we're trying to make our kilograms of drug product per meter square of facility, we think that this mode of continuous and connected manufacturing may actually reduce the manufacturing overall space by up to 50%. And indeed, we're working now with a number of people within the team, including Subhash Chowdhury, who I mentioned earlier on in the slides there, to how those sorts of facilities may actually look. And I think the last point, which is really important, is this sort of cross-industry collaboration to minimize environmental impact. So now starting to partner with the biomanufacturing and technology vendors. And, you know, we're trying to think where we can to shop locally, instead of sort of sourcing these sort of single-use supplies from a sort of much more distant fashion. If we can make these things locally. And what we've done with the Symfonex technology is actually develop and develop that technology in the northeast of England, manufacture that equipment in the northeast of England. We do know that single use is something that offers operational flexibility in the industry, but we also now are looking to think about single use in a reusable way. So perhaps you don't need to dispose them every single cycle or every single batch. And we're starting to move from single use to reuse, but in a way that within the context of that protein. So you still get the advantages of avoiding solvent requirements for sterilization and cleaning, pre-use and post-use, but perhaps we can move towards some reuse in the middle there. And we're generating some data now in a collaborative way with a number of CROs in the region to actually sort of look to see how that's sort of feasible. And then finally, sort of starting to partner again with the technology vendors and the flow path suppliers to look to how we can recycle some of these components. So once they're used, they can be repurposed and reutilized and reformed into other plastics that can perhaps be used in our industry. And there is a strong movement in that. And I'll cite Merck as a large pharma vendor within our industry who've actively started to partner with a number of recycling and waste disposal type initiatives to look to see how they can repurpose some of these plastics that were used in yesterday's process to some of the more non-product contact important aspects like the pallets and things like that. So really, I just wanted to take that sort of 15-20 minutes today to give an introduction to what we're doing and what we're trying to drive innovation to advance our medicines, but in a way that we believe is sustainable. And with that, Ian, I'll hand it back to yourself and happy to take any questions now or at the end of the session.